Good morning, everyone, and welcome to my master's thesis presentation. I looked into seagrass dominance over seaweeds in oligotrophic coastal ecosystems here at CCMAR in Portugal with my supervisors Ana Alexandre, Carmen de los Santos, and Rui Santos. Seagrasses and seaweeds are organisms that occupy the same environmental niche, that is the photic zone of coastal areas. So as they both need to photosynthesize, they compete over the same space. And here on the left, we have the three phyla of algae, green, brown, and red. They can come in different forms and shapes, could be sheet-like filamentous, coarsely branched, thick, leathery, jointed calcareous, or crustose. And in general, they need a hard substrate to grow onto. Whereas seagrasses are plants, so they have leaves, stem, and roots, and like their terrestrial counterparts, they like to grow in a substrate, so that can be sand or more sandy, muddy environment. Depending on the nutrient loading into the marine environment, you have either a macroalgae dominated environment, or whereas if there are low nutrient concentrations, you have a seagrass dominated environment. However, if you have a high nutrient loading into an oligotrophic, so a nutrient poor marine environment, that system topples over. Now, for example, here in the Ria Formosa in Portugal, in Faro, you can observe a lot of seagrasses, and in this oligotrophic environment, it is naturally to observe. So there has been this paradigm that seagrasses dominate over seaweeds in these oligotrophic coastal ecosystem based on their higher efficiency in taking up nutrient concentrations at low concentrations. So the idea is, let's say you have a nutrient pulse, the tide pushes in, stirs up some sediment, introduces nutrients into the water column. These organisms tend to take it up quickly, and for the purpose of this study, we were looking into the kinetic uptake rates. What are these? They are basically described by a saturating function. For a visualization purpose, I put you here the graph on the right. You have the nutrient concentration from low to high on the x-axis, and the nutrient uptake rate on the y-axis. And this describes the Michaelis-Menten model for enzyme kinetics, which states that the initial velocity is equals to the maximum velocity over the half saturation constant Km. And basically what you're looking into, you're testing different nutrient concentrations and you measure the maximum uptake rate. And eventually from different measurements, you get this hyperbolic curve up till to the very maximum that an organism can take, and then it plateaus. And as an uptake normally goes, you have three successive phases, starting with the surge uptake, that is characterized by high and a transient nutrient uptake, then followed by the internally controlled uptake phase, which is characterized by lower, more stabilized and constant rates, and finally, when either the nutrient is depleted in the medium or the organism is saturated, these uptake rates decline. And for the purpose of our study, we were looking for values of maximum uptake rate, Vmax, the half saturation constant Km. And with those two, you can then derive the angle alpha of the initial uptake rate. So the steeper alpha, the higher this value is, you might have an advantage over other organisms. So we phrase these objectives. Are there differences between nutrient kinetic parameters between seagrasses and seaweeds? And if so, can we find differences among surge uptake versus the internally controlled phase? So the rapid uptake versus the stable uptake, among nutrient forms, among taxonomic groups, and still under evaluation among opportunistic versus non-opportunistic species. So based on this paradigm, we phrase the hypothesis that seagrasses dominate over seaweeds in oligotrophic coastal ecosystems due to their higher efficiency in taking up nutrients when they occur. For that, we conducted a literature search following the preferred reporting items for the systematic reviews and meta-analysis protocol called PRISMA, in short, by Moer et al. We had to define keywords for our research in the Web of Science, which we conducted for a time span of 30 years, for journals published worldwide, peer-reviewed and in English only. At the same time, we built a database. So we had a first trial after adding 5 sources to identify which changes were needed. And after a second trial, adding 10 sources, we checked the readability with RStudio for our further and final analysis. So the PRISMA protocol is a guidance of 4 steps. You have identification, screening, eligibility, and included. For the identification part, you have a number of records identified through database searching. In our case, the Web of Science. We had a number of additional records identified through other sources. Those pulled together then underwent screening, which we did by assessing their abstracts to see if they report the values we're interested in. And that was concluded at the end of March. By beginning of April, we started assessing the remaining articles' full text and extracting the values of interest. The keywords we used in the Web of Science were seagrass, seaweeds, or macroalgae, coupled with nutrients, amino acids, ammonium, nitrate, urea, or phosphate, interlinked to uptake rate, Vmax, alpha, etc. For our database, that's an Excel file, including our metadata in the header. We have 3 spreadsheets that are interlinked by a study ID and a unique ID, and the spreadsheets are sources, environmental and experimental. To give you an idea how that looks like, here for sources, the general information about the publication we assess, link the study ID to a unique ID in our environmental sheet, where we register the sample location, the country, the coordinates, the sampling season, which nutrient type was sampled in water column and the pore water, as well as acclimation parameters such as temperature, salinity, light intensity, nutrients used. And finally, in our experimental sheet, we have coupled to the unique ID the species the experiment was done on, so we made sure that the scientific names are up to date. We further refined between species types algae or seagrass, their phyla. We registered the compartment the experiments were done on, that could be leaves, fronds, roots, rhizomes, or the whole organism. And then basically it was up to register our maximum uptake rate and our slope per nutrients tested. I need to mention here, we differentiated between the surge uptake and the internally controlled phase by setting a limit at 60 minutes, meaning everything from 0 to 60. We considered as surge uptake, everything after 60 minutes as internally controlled phase, and that goes for both algae and seagrasses. For our statistical analysis, we initially wanted to go with a 3-way ANOVA, having the species types, seagrass, seaweeds, the nutrients, and the type of uptake, surge uptake, or internally controlled phase. However, due to non-normality of our data and residuals, even after we transformed them, for the purpose of this presentation, we went with non-parametric tests. And I like to mention at the current stage, we're still exploring which model fits best our research question. Coming to the results, for our PRISMA protocol, we identified 1,871 sources that underwent screening. 68 papers remained. We assessed those and extracted our values from 108 studies. And after going through a review, we were left with 104 studies. And that's how our data looks like for the number of articles. Here on the left, the years of publication, starting from 1975 to 2021. And the numbers of studies per country. You can tell the United States are leading with 26 papers, so that makes up 25% of our data set. They can be found throughout the years of publication, but are more concentrated from the 70s to the 90s. I'd like to mention Spain and Portugal emerged more during the last decade, as well as China, which is becoming more and more a big publisher on macroalgae studies. 2, the values of our interest, alpha, the slope, and Vmax, the maximum uptake rate. As you can tell from both graphs, our data is highly skewed. We have big outliers. And for the purpose of this presentation, I will now continue with log transform data only. So here we have the same data. For algae in red and seagrass in green, they were statistically significant for alpha and Vmax, meaning that algae have higher alpha and Vmax rates. Diving a bit deeper into internally controlled phase versus surge uptake, here in purple, we have the statistical significance for alpha and Vmax in favor of algae. Looking at the nutrient composition, comparing amino acid, ammonium, nitrate, phosphate, and urea for both uptake phases, internally controlled and surge, we found statistical significance for alpha during the first 60 minutes for amino acids, ammonium, nitrate, as well for Vmax for ammonium and nitrate, and no significance we found for phosphate. However, here shaded in red, we need to be careful as we only have one value for seagrasses. Interestingly, however, we found the significance for phosphate in the internally controlled phase for Vmax in favor of seagrasses. So here might lie an explanation why seagrasses dominate over algae in oligotrophic waters. And finally, looking at phyla, we have chlorophytes, the green algae, ochrophytes, the brown algae, rhodophytes, red algae, and tracheophytes for seagrasses. Again, in purple, we found a statistical significance for seagrasses versus all phyla of algae in the surge uptake. The same we can see for chlorophytes, so they're statistically significant, different from all other phyla tested in the internally controlled phase for alpha, and we have the same picture for Vmax. Yeah, coming to the last bit for our objectives. Are the nutrient kinetic parameters different between seagrasses and seaweeds? Yes, they are. We could tell. Actually, it's quite the opposite. So we have seen algae are way better off than seagrasses. Therefore, we need to reject our hypothesis. We found differences among surge uptake and internally controlled phase, among the nutrient forms, especially ammonium and nitrate during surge uptake, and for phosphate during the internally controlled phase. We found differences among the taxonomic groups, as you've just seen. And for the opportunistic versus non-opportunistic species, that leads me to my future steps this coming month. So I will need to continue exploring the data set. We still have to decide which statistical model to use. And we further want to differentiate between these opportunistic and non-opportunistic species to see if that might change the picture. And finally, exploring the reasons behind why seagrasses dominate in oligotrophic waters. These are my references for this presentation. And I thank you very much for your attention.